My name is Tom Juenger. I'm an associate professor at the University of Texas in Austin. And we're going to switch gears a little bit. I am trained as an ecologist and an evolutionary biologist. And my lab is generally interested in understanding local adaptation in natural plant populations. And today I'm going to tell you about a really new project in my lab looking at switchgrass, and in particular asking whether there's local adaptation in switchgrass to climatic gradients across its range, and also what the implications of that local adaptation might be for future climate change. My story really starts by acknowledging that climate change is really the environmental concern of our time. Whether climate change occurs, how serious climate change is, what its impacts are for natural environments, biodiversity, agriculture, these will all have really important implications for society. We'll be dealing with this, our kids will be dealing with this, our grandkids will be dealing with this. So with respect to this crowd, we're all pretty familiar with the basic facts and science underlying climate change. If you look at the atmospheric record over the last 2,000 years, we see that there are precipitous increases in several different greenhouse gases, carbon dioxide, methane, nitrous oxide, with a lot of these increases occurring within the last 150 years associated with the Industrial Revolution and the burning of fossil fuels. And so basically these gases act as blankets in the atmosphere, and they increase the absorption of energy. And this translates, we believe, into increases in global temperatures. So this is just a plot of the last 200 years in global mean temperatures. And there's clearly a trend for increasing temperatures. And what's most concerning is that over the last 25 years, there's about a 4 times or fourfold increase in the rate of temperature increases. So these temperature changes themselves may have some important direct impacts. But as a plant biologist, I'm especially interested in some other potentially indirect effects that these temperature changes can have on the climate, and in particular on precipitation variability. So if we look over the last 100 years, we see increases in drought severity as measured by the Palmer Drought Severity Index. And we also see increases in anomalous events, so that precipitation is occurring more rarely, but in larger events. And plants are particularly susceptible to variability in precipitation. Water availability tends to be one of the major limiters of plant communities, plant populations, and agriculture around the world. The fact that fossil fuels are non-renewable and that there's potential of climate change and environmental impacts, of course, we're all interested in alternative energy sources. And one really exciting possible energy source is cellulosic ethanol. And switchgrass, Panicum virgatum, is one promising species for the production of biofuels feedstocks. And so there have been a number of really nice studies in the last few years looking at switchgrass to see whether it's actually economically and energetically viable. And this is a really nice study that comes out of Ken Vogel's group in Lincoln, Nebraska, where they did large farm-scale trials with Panicum virgatum. And at very low inputs of fossil fuels for fuel and fertilizer, they can generate fairly large yields of biomass that then results in a net increase in fuel production. So there are definitely examples of where switchgrass could be a viable biofuel feedstock. But these studies were mostly done in the Midwest under fairly good conditions. And so it's important for us to ask whether or not switchgrass will be economically and energetically viable in marginal habitats, where it wouldn't necessarily compete with crop production for food, and also whether or not switchgrass would be viable in future climates. And again, our notion is that we're going to have one of the major outcomes of increases in temperature will be increases in more severe drought in the future. Okay, so why Panicum virgatum? From my perspective, I'm very excited and interested in Panicum virgatum because it's native to North America. It hasn't been domesticated. It has a very widespread distribution across all of North America. south into northern Mexico. It's very productive, and it's very productive even on marginal lands. It's high yielding, producing between 7 and 10 tons per acre. In general, people can grow it with very low inputs. It's perennial, so a farmer can plant a crop and harvest it year after year for 10 to 15 years. From my perspective, it's also very interesting because it has a long history of agronomic work that is a nice foundation for some of the things that we'd like to do down the road. When we think about the natural history of Panicum virgatum, it has several different features. There are clearly two different potentially adapted types, lowland types and upland types. The lowland types tend to be the most high producers, the high yielders. They grow well in mesic environments. The upland types are much more conservative in their growth. They produce less biomass, but they're more tolerant to abiotic stresses. There's a lot of complexity to Panicum virgatum. It has a very large genome. There's polyploidy. Most of the accessions, the lowland accessions are tetraploid. Many of the upland accessions are octoploid. Ed Buckler's group has done quite a lot of work looking at ploidy levels. There's clearly aneuploidy that's very common as well. JGI has shown some interest in Panicum virgatum by generating EST resources, back sequencing, and some genomic sequencing. And these are tools that are going to be really important as we start to think about developing Panicum virgatum as a potential biofuel crop. But I want to say that sitting here and hearing everybody's talks, it's a little bit disturbing because Panicum virgatum is very far from being a model system. We have very, very few tools and resources, and it's going to take us a long time to develop those resources to make progress. So from my perspective, it's a very interesting system because Panicum virgatum occurs across really remarkable climatic gradients. It spans over 8 USDA cold hardiness zones and up to 60 inches of precipitation variation east and west. And so the type of questions we're interested in are, is switchgrass locally adapted to climatic gradients, and at what spatial scales? Which gradients are most important, either these temperature scales or precipitation patterns? What are the mechanisms, traits, and genes that might be important for current or future adaptation to climate? And ultimately, what we'd like to be able to do is to try to predict or forecast how switchgrass will perform as a biofuel crop under future climates. And just as a little cartoon example of how we might try to do this forecasting, we've been working with the Almanac crop modeling system. And Almanac is a crop modeling system that we can parameterize particular types of genotypes. And in this case, we've mostly worked with the Alamo cultivar. The Alamo cultivar is one of the highest yielding, most widely planted cultivars, switchgrass. And it makes yield projections in the context of soil characteristics, both water and nutrient balances, climate, and then plant physiological characters that we've actually measured on the stand level. And what we've done that's a little bit different is we've tried to port together the Almanac crop modeling approach with climate predictions of the future. And so we've been using downsized IPCC climate scenarios for Eastern North America, looking 70 years out. And you can see from these panels, these are actually results from simulations. This top panel corresponds to current predictions of yield. And we can actually validate how well our model is predicting based on field trials that people have completed with the Alamo cultivar. And then the second panel is really the middle-of-the-road climate scenario where there's some reductions in CO2 emissions. And then the bottom panel is associated with the worst-case scenario. And what you can see is that much of the west and southwest really tank under even the middle-of-the-road climate predictions. And so that suggests that switchgrass may not actually work out so well as a biofuel crop species on marginal lands in the future. But this approach fails in a number of ways because it doesn't incorporate genetic variation within switchgrass, the potential for adaptation or breeding, or improvements in management. And likewise, it doesn't really incorporate the uncertainty associated with these climate modeling approaches. Okay, so I think these are real failings because we know that we have really good evidence that there's local adaptation and lots of genetic variation in switchgrass. Going back to the 50s, there were some really wonderful experiments completed by Calvin McMillan. Calvin was a faculty member in Lincoln, Nebraska, and when he started his career, he drove around North America and collected accessions of lots of the dominant grasses in North America, including Panicum brigatum. And he grew them at several common garden sites and then measured a whole host of traits in those plants. About halfway through his career, he moved from Lincoln, Nebraska to Austin, Texas, where I go to school. And in Austin, he also developed common garden experiments. And so he could compare in these common gardens the impact of the environment and the impact of genetic variation on a variety of different plant characters. These little dots correspond to actual locations where he collected plants, and these figures just show the characteristics of different clonal lines at different sites. So just as an example of how much variation he saw in his experiments, going from coastal Texas to panhandle Texas, he found over 3 months of genetic divergence in flowering time. So an enormous amount of genetic variation, standing genetic variation in Panicum brigatum. And interestingly, a lot of the genetic variation that Calvin observed was geographically structured or associated with climate gradients. So here's just 2 panels showing a really strong correlation between latitude and flowering time, one of the traits that he studied, in both Lincoln, Nebraska and Austin, Texas common gardens. And so you can see that southern types flowered late and northern types flowered early, both in Lincoln, Nebraska and in Austin, Texas. And then following Calvin's work, there was a number of studies funded by USDA and DOE where researchers went out and collected accessions of Panicum brigatum and grew them both at home locations and away locations across North America. And again, generally they have found that plants perform better in their local habitats than away habitats. And this notion of local adaptation, maybe at the scale of plant adaptation zones, has been one of the main drivers in breeding and agronomic practices in Panicum brigatum. Okay, and so I think that there's really good evidence for local adaptation, at least on the scale of ecoregions in North America. So our project really takes off from where these previous studies ended with common gardens by trying to add some more mechanistic understanding of how plants perform well in particular habitats and adding a genomics component that we hope ultimately will feed into improving switchgrass for biofuels. And so we have 13 different common garden sites that we set up this last year, ranging from South Texas to up in Missouri. And at each of these sites, we're planting a collection of plant material, including northern and southern types, tetraploids and octoploids, and lowlands and upland ecotypes. We're also focusing at 3 sites in central Texas on the role of drought in plant performance. And the way we're doing this is using experimental rainout shelters. And so these are basically large greenhouse structures that exclude rainfall. And then underneath these rainfall structures, we can irrigate in various ways to mimic future projections of precipitation. And so for central Texas, there are predictions of about a 20 to 25% decrease in the magnitude of precipitation each year, and then changes in the timing of precipitation. And so we're manipulating those in randomized block designs. So these are long-term investments. These plants will be around for 10 or 15 years. And so we're just getting started with these experiments, and we're going to be measuring a whole host of different traits. My interests originally lie in plant physiology, and so we're very interested in C4 photosynthesis and how sensitive it is to drought. We'll be making lots of gas exchange measurements, looking at photosynthesis and transpiration and water use efficiency and leaf level plant performance and status. We're also very interested in nutrient use under drought and nutrient mobilization in the context of harvest. So some of the postdocs are looking at nitrogen and nitrogen mobilization across the plants. We're looking at biomass characters, phenology, and then from a genomics perspective, we're trying to get transcriptome data that allow us to start to understand how locally adapted types might be responding at a molecular level. So much of our understanding of plant responses to abiotic stresses stem from transcriptome studies. And at this point, we have none from Panicum virgatum. And so we'll be able to focus on transcriptome responses to drought events, to recovery following precipitation, and long-term acclimation. So the approach that we're trying to develop is a tag sequencing approach using next-generation sequencing platforms. And the strategy is really simple. We're harvesting total RNA, enriching for a poly-A fraction, and then basically having a library prep that grabs a chunk of 3' sequence, barcodes it so that we can pool all the barcodes together, and then we're sequencing on the solid sequencing platform. And then we take those sequences and filter them, and then we're actually trying to map them using shrimp to some EST resources that JGI has generated in collaboration with Noble and Mike Urdavardi's group for the Alamo AP13 genotype. And there's actually a poster about these EST resources, poster number 55. And so ultimately, these data sets will basically look like a stack of tags that we observe for different transcripts. And we can count up the tags and then make direct contrasts under control and manipulation treatments. So this is relatively new technology, and we're trying to use it in a very, very complex genome. And so we spend a lot of time trying to work through some of the technical details. And two technical details that might be very important are the read lengths of the tags and the depth of coverage of the sequencing. And so we've been working on the solid platform and primarily using 50 base pair tag sequences. And we can do a little bioinformatics trick, just trim off some of the sequence to look at how sensitive our mapping is to the length of the tag and the uniqueness of the ESTs. And so we've done that game, and it's pretty clear that tag length seems to not matter enormously. We get very similar tag counts and expression measures using either a 50 base pair read or a 35 base pair read. With respect to depth, we've sequenced some libraries very deeply, and what we tend to find in this rare fraction curve is that it asymptotes pretty quickly, such that if we sequence a library to 2 or so million reads, we get 80 to 90% of the tags detected in a leaf sample. And so we're just getting started with this. We've done some pilot experiments, and the pilot experiments basically are greenhouse potted plant experiments using the wild-type line Alamo AP13. We're growing these plants in gallon pots, and then over a period of a week, drying them down to some pretty severe stress levels. And along the way, we can make repeated samplings of physiology and RNA for transcriptome studies. So I only have one small little experiment to show you, comparing pre-dawn wet and dry plants or midday plants, wet, dry, and some plants that had actually been rewatered. And these are very, very severely stressed plants where they have leaf water potentials of about -4 megapascals. So pretty stressful conditions. So under this scenario, we've detected about 25,000 unique tags from leaf tissue. And we've been using a Poisson-based general linear model approach for count data using the software package EdgeR, which is actually a really nice software package for this type of count data. So here's some of the results. This is a smear plot that basically describes the variability in the data. On the x-axis is the concentration, the average concentration of tags in the sample in the libraries. And on the y-axis is just the log fold change difference across, in this case, the wet and dry treatment, where the red dots are actually genes that we detected as differentially expressed at a really stringent false discovery rate. So at least in our first experiments, the data looks very, very promising as a tool that will allow us really high throughput transcriptome studies in Panicum virgatum. Right now, we're paying about $100 a sample, which is a really, really great price for a whole genome transcriptome analysis. So with respect to this pre-dawn data, we detected over 1,000 differentially expressed genes at a really stringent FDR. For the midday data, we detected almost 2,000 differentially expressed genes. Many of the expected functions from other previous studies in plants. We see lots of changes in redox regulation, some genes that are probably very important in osmotic adjustment, something that we're very interested in from a physiological level. In terms of plants that have been rewatered, we see lots of photosystem genes and transporters, and there are a whole host of interesting transcription factors. But really, I can't tell you much about the biology yet, in part because the data is very new, but also because we have so few resources in Vergatum that annotates the genome. So annotation is going to be a huge problem for us. It's a huge problem from everybody, I believe. Likewise, this is all with one genotype. And what we really ultimately want to be doing is making contrast between locally adapted different genotypes. Many of them have different ploidy levels. And so we need to start incorporating the diversity that will come from having multiple copies, gene families, and likewise genetic diversity. So from some of the sequencing that JGI has completed, we know that Panicum Vergatum has a lot of genetic diversity. And if we're looking at a 50 or 60 base pair tag, we have to be very concerned about the role of sequence polymorphisms and the mapping efficiencies of the data. So still some real challenges ahead of us, but I think we're optimistic that this will give us at least the first tool to learn a little bit about how the plants respond to stress and then be able to make some comparisons to some of the other grass species. Okay. So these common garden experiments and studies of traits may let us make some hypotheses about mechanisms, but really what we want to do is find the genes that underlie local adaptation. And the only way we're going to do that is by doing some kind of genetic mapping. And so we've started outbred QTL mapping with Panicum Vergatum using a mapping population that was generated by Christian Tobias, a USDA researcher. He crossed the Alamo line with the Canlo line, a line from Texas and a line from Oklahoma, and created about 200 full sibs family that he shared with us. Christian genotyped these at a huge number of microsatellite loci to create the first good linkage map of Panicum Vergatum. And what he discovered, which was very exciting, was that although they're tetraploids, they show disomic inheritance and preferential pairing. So it looks like Panicum Vergatum is a diploidized, in this case, tetraploid. So Alamo and Canlo, they're both agronomic cultivars. They've experienced some breeding. They're both high yielders. We've planted this under a rainout shelter and actually have started collecting some data from these experiments, and we're actually mapping QTL. So I think for the first time, we're actually identifying pieces of the genome that impact biomass accumulation in Panicum Vergatum. These are for early tiller production. We map 2 very big intervals on Panicum Vergatum chromosomes that affect tiller production, and 1 QTL that's derived from the Alamo parent. But again, this population wasn't designed for looking at local adaptation, and as such, we've been making some of our own crosses based on some new plant material. And so we've been collecting lots of new germplasm, in particular across climatic gradients. One of the postdocs in the lab, David Lowry, has been working hard at making crosses. And so, for example, we've been crossing lines from central Texas near Austin with lines from very drought-prone western Texas. Given that these are outcrossed plants, we have to make complex pseudo-test cross designs. So we take 2 parents, make an F1, cross those F1 to generate an F2. It's going to take us a while to generate the markers and new linkage maps and get these plants in the field. But I think in the coming years, we'll start to be able to map loci that might relate to adaptation across these climatic gradients. So again, Panicum Vergatum is a really important and exciting species, but it has a number of limitations, including the large genome, the fact that it's an obligate outcrosser, and that it has polyploidy. And so we've been looking at a couple of other species that might be interesting models that we could use both in a genomic context and also for understanding local adaptation. And so this is a phylogeny of the panicoid subfamily based on some chloroplast sequences. And you can see here's the position of Panicum Vergatum, the agronomic switchgrass. Foxtail millet is our closest relative where we have a full genome. It's shared a common ancestor with Panicum Vergatum about 13 or 14 million years ago. It actually is very, very different in its growth form. It's annual, not perennial. It's very different, and so we'd like something closer. And we've been looking carefully at this little clade, the Panicum diffusum clade, and in particular Hall's panicum, as a potential model species. So Panicum Holly is diploid. It has 18 chromosomes. It has a relatively small genome, about 500 megabases in size. It has a mixed mating system, but when we bring it into the growth chambers and greenhouses, it seems to be predominantly selfing, which is going to be really great for breeding applications. We can get it to flower between 6 to 8 weeks from seed to flower. We have both upland and lowland types, and so there's clearly some local adaptation. And it's very common across this very steep precipitation gradient in Texas. There are two subspecies Hollyi, that seems to be really adapted to xeric conditions, and subspecies Philippes that's more limited to mesic areas. So we'd like to ask very similar questions to the story I just told you with Panicum virgatum, but also with Panicum Hollyi. And here's just some pictures of the different habitats, very wet grassland east of Austin, 6,000 feet in the Davis Mountains or Texas Sky Islands, in the Chihuahuan Desert in Arizona and New Mexico. And so since we can self this, we've been making crosses between parents that we've collected across the climatic gradient. We can take these F1s and self them to make F2 populations, and then self those F2 populations to make standard recombinant inbred lines. And then the idea is to plant these recombinant inbred line populations across the climatic gradient, and perhaps under rainout shelters as well. But what's really exciting is JGI has done a small bit of pilot sequencing, genomic sequencing, including 454 paired end and standard reads, and Illumina paired in reads. Tried to get a little bit of data to learn more about the Panicum genomes. And we've been trying to assemble these reads. We've basically taken a hybrid strategy where we take the Illumina reads and assemble them using SOAP de novo, and assemble the 454 reads using Nubler, and then we're merging those assemblies. And although we're just really playing around at this point, just this quick assembly, we have about 500 megabases of the genome that's covered. That's about 91% of the genome at 50x coverage. And of course, the issue is that all of this assembles into very, very small little reads. Over half of the assembly is in bricks the size of less than 5 kb. And so like all people studying plant genomes, we're struggling with how to assemble these very large, complex, repetitive genomes using next generation sequencing. But one thing I want to emphasize is that even these little bricks have been incredibly helpful for us in developing genomic tools. So we can already take these assemblies and annotate them for gene discovery. We've also been using these small little bricks to validate high-throughput genotyping schemes like RAD approaches that we've taken. And a postdoc in the lab, Eli Meyer, has been a really helpful super guy leading all of our next-generation sequencing work. Okay, so to conclude, climate change is a very real concern, especially its direct impacts on agriculture. We really need to plan for this change when anticipating biofuel productivity, especially in the context of drought on marginal lands. There's really good evidence for local adaptation in switchgrass, and I think, as Ed argued earlier, this functional diversity will be really critical for switchgrass breeding and improvement. But the problem is we have all of the issues that Ed described. It takes us very long to cycle any breeding of Panicum virgatum, on the order of years. And yet we really need to improve this crop quickly. And so I think that genetic and genomic tools and perhaps genome-wide selection, like Ed described, will be really critical and important for our chances for improving switchgrass for biofuels. And so I'd like to first just thank our funding sources, NSF Plant Genome, JGI for their interest in sequencing some diploid panicums, and also the Texas Advanced Computing Center for computational time for all of our assemblies and mapping. And then a number of collaborators, co-PIs on my grant, Christine Hawks, Tim Kitt, and Phil Fay, USDA collaborators, Dan Roxar, Carrie Berry, and folks from the Noble Foundation. I'd love any questions and comments. Thank you.
Pretty simple question. Is there any chance there's a diploid switchgrass out there?
So Christian Tobias has, I think, discovered some dihaploids from crosses. They're very sick. They're not happy plants. Yeah.
So when you've done the climate digging, has that all been with essentially short read inserts? Because actually, I'm pretty impressed by your numbers, if that's all.
Yeah.
Yeah.
You know, I mean, it's as hard as it can get in the sense that we have heterozygosity and not a ton of
But
coverage.
these are all paired in, 500 base pair reads?
We have a diversity of insert sizes. They're all pretty small.
But no 2 KB ones?
Not yet, yeah.
Yeah, I mean, I suspect, you know, I've seen some work in maize on Mo 17, throws them 2 and 5 KBs. I think that'll snap.
That's our hope.
Very nice.
Our second hope is that we can try to couple together our linkage map in real populations and use that to help assemble some of the rigs. Yeah. Yeah.
Tom, can you sort of compare and contrast for this, those of us who don't really know these things, Panicum capillary and the one that you just advocated?
Sure. Yeah,
so capillary is a small diploid, right?
Yeah, so Panicum capillary and Panicum Holly are both members of the same small clade. There are 12 species in the clade. It's called the diffusum clade. They all have relatively small genomes. Panicum capillary is a diploid. It's an annual, a weedy annual that occurs across much of North America. And in contrast, these are perennials. They're more vigorous, larger in size, and more restricted in their range distribution.
It's a diploid?
Yeah, capillary is a diploid as well. Hollyi is a diploid as well, yeah.
Okay, well, thank you very much.